So good evening, all. It's really awesome to be back here, especially as Alex mentioned, there's so much noise around us these days, and it's so wonderful to come here, and hopefully we have a few days of immersive geeky science and chats. So kicking this open, it's the first session, Immunology Power, and also Power to Immunology. And so Amita and I are co-chairing this session. So first, to kick it off, I would like to, it's my honor to introduce Bali Pulendran from Stanford. So Bali, as many of you know, has been at the forefront and a pioneer in systems immunology for more than 2 decades. And he's a regular. And last time I think he came to the meeting was during the pandemic. We have to hear him over Zoom. But this time you got the real Bali, physical, in person 3D. So.
No, that's fine. Thank you very much. Well, John, thank you so much. And I'd like to thank the organizers for inviting me, and I'm really honored to be here, especially at this time. And I think you've named the first session very appropriately, Immunology Power, because it is all about vaccines, and they are the power of immunology. So what I want to tell you in the next 20 minutes, and I promise it will only be 20 minutes, is some work that we've done to understand the human immune response to vaccines. Now, if you're in public health, vaccines are, of course, the most important public health tools. They save millions of lives, but if you're an immunologist, they are incredibly useful probes on the human immune system. Why? Because we all get vaccines, and you can use vaccines to perturb the human immune system in a very synchronized way. If you will, it's a stress test on the human immune system. If you're a cardiologist, you would have people run on treadmills and hook them up to an EKG. But if you're an immunologist, you would vaccinate people to stress test the human immune system. And then if you apply all the tools of systems biology to understanding the nature of the innate and adaptive response to vaccines, this really gives you a wonderful glimpse into the workings of the human immune system. And of course, we humans are so diverse, genotypically, phenotypically, and what are the molecular networks that are manifest in this diversity, and what are the factors that control this diversity? Is it host genetics? Is it the microbiome? Are there environmental triggers? All of these very fundamental questions in human biology could be addressed by using the most practical and most boring tools that we call vaccines. So this dichotomy between translation and discovery in this field disappears, because translation is discovery, and discovery is translation. So for us, the journey began about 2008, when we did this experiment in human immunology by using yellow fever. Yellow fever is a live virus vaccine. It was made some 80 years ago, and it's incredibly potent at stimulating long-lasting immune responses. The efficacy is 99%. And so it was a live virus, single-stranded RNA virus. Nobody really knew how it works. So we thought that this would be a terrific model to begin to understand the immunology, the immunological basis of this. And what we did is shown here in the slide, where we vaccinated healthy humans, collected blood samples at all those time points shown there, and then did a deep profiling of the innate and the adaptive response to vaccination. We looked at the adaptive response using tetramers. This is Rafi Ahmed's work to look at the antigen-specific T cells. We measured neutralizing antibody responses. We also measured the signatures, the transcriptional signatures induced by vaccination. And computationally, we could define an early signature in the blood that correlated with the later immune response, and also use machine learning to now predict the magnitude of the T cell response and the neutralizing antibody response in a completely independent trial, trial number 2, shown on the right-hand side. So at the time that we were doing this work, a completely independent study by the group of Rafiq Sekali and colleagues, who is here in Montreal, did a virtually identical study, but completely independently of ours. And it was a sheer delight that both of these studies converged on the same pathways, same transcriptional signatures that were associated with and predictive of the immune response. Since that time, this field has really sort of mushroomed. Lots of people, many in this group here, working on this. Our own work has sort of progressed. I just want to highlight a couple of things. One thing that we were very keen to do was to go beyond just transcriptomics. Now, could you now use metabolomics, high-dimensional flow cytometry, epigenomics, and so on and so forth to construct a multi-scale response network to vaccination? And in this paper that we published back in 2017, we did precisely that, where in response to the shingles vaccine in humans, we were able to construct what's called a multi-scale, multifactorial response network computationally, and that network is shown there. And what's always fun with these kind of activities is the richness of the biology. This is merely the starting point, but to go from this point to understanding biological mechanisms that drive and sustain vaccine immunity is so much fun. And it would be remiss of me not to highlight this paper, these two papers that were published as part of the HIPC consortium. I know most of you have heard of what HIPC stands for, HIPC. It's an NIH-funded consortium. It stands for Human Immune Profiling. And a lot of people in this room are part of that consortium. We're very lucky to be part of that consortium. These were studies that were led by the HIPC consortium, where they looked at 3,000 samples from 820 humans who had received across 28 different studies, who had received 13 vaccines. And the purpose of these studies was to ask, is there a universal predictor of vaccine antibody responses across all vaccines? And in this first paper, what we could show was that there was a plasmablast signature. Plasmablasts are antibody-producing cells that are induced by vaccination, typically at day 7, but not always at day 7. This signature was highly predictive of the peak antibody response. And in this other paper that was also published in the same journal, led by Steve Kleinstein and Rafiq Sekali and others, they could show at baseline there were signatures that were associated with the antibody response. But with that intro, what I really want to highlight to you in the remaining 15 minutes are two issues, two topics, I think crucially important areas, fundamental questions, where we think this sort of systems vaccinology work is beginning to shed some light. The first one is the vexing problem of the durability of antibody responses. This is a problem that we are all personally affected by, because when we get a vaccine, we'd like to make sure that the vaccine protects us for quite some time. Sadly, that's not the case with the seasonal flu shot, which is not very long-lasting, or some of the mRNA vaccines. The second issue is what we have termed, in this rather catchy sort of phrase, integrated organ immunity. Put simply, it's asking a simple question: what do you need to protect you against an infection? Is it B cells, is it T cells, is it innate, or some combination of those? And what can we learn about the synergy that happens between these three different elements of the immune system? And more importantly, how could we harness this synergy to make what we call a universal vaccine that conceivably could protect you from anything? And that sounds like a pipe dream, but I'd like to share some of the data with you. So let's begin with the first part, durability. What do we mean by durability? You have to be fairly precise in this definition, so I'm going to focus on the antibody response. If you look at Charlie Janeway's immunology textbook and you look at the kinetics of an antibody response, it would look something like this. Vaccination causes an increase in the antibody titers. This is followed by a rapid exponential decay phase, followed by a much more gradual, slower, linear decay phase. Now, consider what happens when you give a human a different vaccine, a red vaccine. You see immediately that the peak titers are considerably lower, but oddly enough, the magnitude at the durability of the response seems to be similar to the green vaccine. You see that horizontal dotted line is a threshold of antibody concentration that you would need for protection against infection. Or to put it in more technical terms, that is what we call the correlate to protection. Now, consider what happens to a blue vaccine. The titer is high, but the durability is very similar. Why is that so? Immediately you'll recognize this is so because the exponential decay is so very different between these vaccines. So this tells us that if you wish to understand the durability, that you need to deconstruct this complex phenomenon into its constituent parts and solve each part separately and ask what are the immunological factors that are critical for each of these phases. Now consider idealistically, if you wish to design a gold standard vaccine, and let's call it yellow for yellow fever, this would look something like this: high peak titers, gradual decay phase, both initially and also the subsequent decay phase. Now, mathematically, you might say the durability at time t is a complex function of the peak antibody titers multiplied by the rapid decay slope by then the slow decay slope. So for the purposes of this talk, I want to focus on what happens, what are the immunological factors that control that initial rapid decay phase. Now, this audience does not need this slide. It's basically to say that activated B cells, some of them go inside a germinal center where they can, if they survive, they can now emerge as memory B cells or long-lived bone marrow plasma cells, which are the cells that maintain the antibody response for years, sometimes decades. What I'm going to show you, some of what I'm going to show you is published. It's in this paper that came out just a couple of months ago. So we use the tools of systems biology to begin to define early transcriptional signatures that could be induced by vaccination and that were associated with the durability of the antibody response. The vaccine that we looked at, as shown here, it's the H5N1 bird flu vaccine from GlaxoSmithKline, given by itself or with this adjuvant known as ASO3, 2 shots, day 0, day 21. Much to our surprise, what we could see was that there was a transcriptional signature. We're looking at this heat map. We're looking at these blood transcriptional modules that are sets of genes that are co-regulated. Oddly enough, what we saw was that there was a gene set associated with genes expressed in megakaryocytes and mRNAs contained in platelets that were highly associated with the durability of the antibody response to this vaccine. This was a bizarre finding because, of course, platelets and megakaryocytes, as far as we can tell, have not been reported to have any function in terms of the vaccine responses or antibody responses. To dig deeply into this, what we did was single-cell RNA sequencing, and we could see that indeed there was a cluster of cells shown here in the green circle that looked like platelets. Why? Because they didn't express any of the genes that you would be expected to express in lineage-positive cells, but rather they express many genes that are contained in megakaryocytes. Now, there were also clusters of cells shown here in red that appears to be doublets between platelets and monocytes and other cells. So just for the non-aficionados, what are platelets and where do they differentiate from? Well, of course, platelets differentiate from megakaryocytes. Platelets themselves do not have nuclei, but they do contain RNA in their cytosol. Why is it that they have RNA in the cytosol? Because they differentiate from these large, beefy, multilobulated cells called megakaryocytes that are present in the bone marrow. And of course, these cells have an abundance of cytosolic messenger RNA, and when they differentiate, so platelets carry some of the messenger RNAs with them. So what controls the differentiation of platelets and megakaryocytes? Well, it's this cytokine called TPO, or thrombopoietin. Thrombopoietin binds to this thrombopoietin receptor that is expressed in hematopoietic stem cells, but also on megakaryocyte erythroid progenitor cells. And so what we asked was, well, what would happen if you inject thrombopoietin into mice to perturb the differentiation of platelets and megakaryocytes, and then vaccinate these mice? What impact does this have on the immune response? That's the experiment we did in mice. We immunized with recombinant spike protein with the ASO3 adjuvant, boosted it 21 days later, but around the time of boosting, we injected thrombopoietin into these mice, and you can see on the right-hand side, these are the antibody titers. In gray are mock immunized mice, so they did get the vaccine, but they didn't get TPO. In blue are mice that received the vaccine with the TPO, and Y is a log 10 scale, the y-axis, and you can see there's a substantial increase in the titers. That seems to be sustained at least for 2 months after vaccination. So let's do the converse experiment. Now let's knock out the receptor for thrombopoietin from mice and say, what is the impact of this on the immune response? So there we are. If you look at the left facts panel and then the plots next to it, you can see the percentage of megakaryocytes in the bone marrow, and the absolute numbers of megakaryocytes in the bone marrow is substantially reduced, although not all the way to zero, but substantially reduced. The antibody titers in these mice is reduced, and then the frequency of bone marrow plasma cells is reduced as well. And so finally we asked, how is it that these cells that are present in the bone marrow are having such an effect on the antibody response to a vaccine? So we hypothesized that there may be some sort of cell-cell communication in the bone marrow between megakaryocytes and these plasma cells that I mentioned to you before. So to address this, what we did was to isolate bone marrow plasma cells from humans. We isolated megakaryocytes from the same human. We activated the megakaryocytes with thrombopoietin in vitro and co-cultured them. And then we looked at the survival of the plasma cells, but also the antibody secretion shown there in the middle panel and the one to the right. And you can see that the survival was greatly enhanced, as was the immunoglobulin production. So it appears that megakaryocytes are providing some critical survival signals to these bone marrow plasma cells and enables their enhanced survival and antibody production. But what survival signals are they producing? So we screened a whole bunch of different molecules, some of which we knew were expressed by megakaryocytes. And the first thing is that you absolutely need beta-1, beta-2 integrin, and that's not surprising because cell-cell contact requires that. We also needed interleukin-6, APRIL, and this MIF. MIF stands for migration inhibitory factor that's produced by megakaryocytes that binds to CD74 in plasma cells. All of these factors seem to be critically important in this. As we were submitting this paper, we were delighted to see this classic paper that was published in Blood, in which the group of Rudolf Mans and Ian McLennan had shown in mice that when they did confocal imaging in bone marrow, there was this juxtaposition of the megakaryocytes with plasma cells. So to conclude this part, basically the model we have currently is that vaccination causes a signal, we don't know what signal that is, that results in activation of megakaryocytes. Of course, vaccination induces activated B cells to form plasma cells, which traffic everywhere, including the bone marrow. And in the bone marrow, these plasma cells have a lease on life that enables them to survive longer and therefore contribute to long antibody production. Finally, I just want to say that the signature we had defined initially with the H5N1 avian influenza vaccine and the seasonal flu vaccine, we could also test this with a number of other human vaccines, and these are shown here, including the pneumococcal vaccines, meningococcal vaccines, and in the majority of these vaccines, we were able to use this signature to predict the durability of the immune response to these vaccines. So, John, I have 3.5 minutes. Let me summarize quickly the second part, and it'll be a very abbreviated version, all right? So, but this would dovetail into what you're going to hear from Jeff and others. The idea is that the human immune system has evolved many different mechanisms, and vaccines or vaccinologists have focused on the antibody response, but really, I think we need to revisit this, and we need to look at this as a system. It's a systems immunology meeting, and how these systems come together to create protection. Now, you all know about trained immunity. This was work pioneered by Mihai Netia and colleagues that say that the innate immune system has a form of memory. We think that this is manifest through epigenetic changes, and I think Musa is going to talk about that. And this is a remarkable feature, unappreciated feature of the innate immune system. What we had seen just a few years ago was that this was indeed the case with vaccines. If you vaccinate humans and you do single-cell ATAC-seq, weeks or sometimes 2 months after vaccination, in monocytes in the blood, we saw these remarkable and persistent epigenetic changes. What were those epigenetic changes? Well, the chromatin loci targeted by certain transcription factors such as IRF1, 2, STAT1, STAT2 were wide open for at least 2 months. But in the same monocyte cell, chromatin loci targeted by AP1 transcription factors such as CFOS and CJUN were tightly closed. And this had a functional consequence because if you isolated these monocytes after vaccination and exposed them to a completely unrelated virus such as dengue or Zika, not the virus against which the vaccine was intended, these monocytes were able to control virus replication. And this suggests that this epigenetic program that's induced by vaccination in these monocytes that lasts for 2 weeks is an antiviral program. It is virus agnostic. It seems to be able to control virus regardless of their antigen specificity. Now, we've extended the study to looking at a whole bunch of different viruses, vaccines, to ask, are there certain patterns of epigenetic imprinting depending on the kind of vaccine? What happens if you use an mRNA vaccine, a live viral vaccine like yellow fever? What I can tell you is that there's this remarkable structure to the epigenetic changes in monocytes. This structure is shown here in this simple slide. Basically, we're looking at classical monocytes in the blood in response to vaccination with 6 different types of vaccines and asking what are the epigenetic changes using single-cell ATAC-seq, and what kind of patterns are these? Do some of these changes co-cluster with others? Are others mutually exclusive, others reciprocal? What you can see in this heat map is that there's a reciprocal nature. We hear the word reciprocal when it comes to tariffs, but here it's sorry, but it is, because the IRF targeted loci, when they open up, the AP1 targeted loci close up, you see. But on the other hand, the IRF targeted loci are opening up at the same time as the CCAT enhancer binding protein targeted loci. So there's a basic structure to these changes, and we call them motif superclusters, epigenetic superclusters. We're probing the functional consequences of these. And lastly, what I want to leave you with is to say it's not just epigenetics, so we've super focused on epigenetic changes in one cell type, monocytes, but surely the immune system has so many different cell types. Surely trained immunity could possibly be manifest by the interaction between all these different cell types. Why should we exclusively focus on epigenetics in a single cell type? And so the gist of this is that what we call integrated organ immunity. This began with our work on this BCG vaccine. Audrey Lee, a graduate student here, did some experiments in mice where she gives the BCG vaccine intravenously, and then several days later, 7 days, 14 days, 21 days, 28 days, she infects these mice with SARS-CoV-2 and sees this dramatic effect in terms of protection, but there's a window of protection. You see, at 7 days and 14 days, there's no effect of BCG on protection against SARS-CoV-2, but at 21 days, 28 days, 42 days, there is, but even at 3 months. This is even more dramatic when you look at the quantity of virus, SARS-CoV-2 virus, in the lungs. You see, at 7 days, there's no effect, 14 days, there's no effect, 21 days, there's a 93-fold drop, 28 days, 435-fold drop, 42 days, 92-fold drop. But that is only in the lung and not in the nose. So it doesn't prevent infection through the nose, but it does prevent dissemination to the lower respiratory tract. How is this happening? It turns out T cells. The T cells that are induced by BCG is feeding back on the innate system to keep the innate response alive. How do we know that? We did spatial transcriptomics. This is a whole lung of a single mouse, unvaccinated. In green, you can see the epithelial cells. In pink, you should see CD4 T cells, but you can't see very many. Why? Because it's an unvaccinated mouse. What happens when you vaccinate the mice a few weeks later? There's this massive infiltration of T cells, CD4 T cells, and what genes do they express? They express lots of interferon gamma. And what happens in a third group of mice if you vaccinate and get rid of the T cells? How? By injecting an anti-CD4 antibody. So clearly you've eliminated the cells. Now let's ask, what is the impact of these T cells on the innate signature in epithelial cells and other innate cells? So you can see this on this heat map. These are the top genes that are expressed by these epithelial cells. These are the targets of viral infection. On the left panel, BCG switches on lots of interferon-stimulated genes. It's an antiviral program. Now, if you deplete the CD4 T cells in BCG immunized mice, you don't find that antiviral program. So it's a feedback of the CD4 T cells. And you can look at your favorite genes and do the numbers. So this is my last slide. Basically, what I want to say is that as you look at this issue of trained immunity, you have to have a wider perspective beyond epigenetics and look at the immune system as a system. That is a message for the Systems Immunology meeting, that it goes beyond a cell or a single cell. I know we are enchanted by single cell technologies, but you really have to look at the physiology of organisms at different hierarchies, not just at the single cell, at the organ level, at the organismal level, and so on and so forth. And what this data illustrates is that a feedback, there's a synergy between the T cells feeding back on the innate system to keep the innate system alive. And so this is what I wanted to say. Thank you for your attention. And I think I already referred to, so the people who did the durability work are Thomas Hagen, Mario Cortez with Nadine Rufael, and the people who are doing the integrated organ immunity work are Audrey Lee and Haibo Zhang. Thank you very much.
We have a couple of minutes maybe for a quick couple of questions. Yes.
And great talk. I was wondering what is known about vaccine response in people with, say, thrombocytopenia or some kind of mutations in these platelet-related genes.
Yeah. That's a good question. People with thrombocytopenia, as you know, have a lot of defects in terms of their megakaryocytes and so on and so forth. Some of them are on TPO, so I don't know if there's data on the vaccine responses, but what we're thinking is a study in healthy humans, sort of a small phase 0 study, who do get thrombopoietin and then get the vaccine just to see what the impact is. But we don't have data on that yet. Yeah.
Okay. One last super quick question over. Sorry, Rafiq, there's a hand behind you first.
I was wondering, like, you kind of proposed like CD4 is kind of controlling the innate immune response, but is that not like controversial to what textbook immunology kind of tell us, like innate immune response is first and kind of triggering the...
Yeah, that's a great question. I think the lesson is don't ever believe what you read in textbooks. No, you're right, absolutely. I think the innate system absolutely controls the adaptive system, but why should it be a unidirectional thing? There's reciprocal, as I say. Okay.
Okay, great. Thanks, Bali.